Dr. L, and this is the second part of the transport lecture focusing on proteins. So when you're looking at the proteins that are involved in active transport, there's some additional categorization or adjectives we can use to describe them based on how they function. And they're shown here: uniporter, symporter, and antiporter. A uniporter carrier protein will transport something in one direction across the protein, just one thing. A symporter will transport two things at the same time, and an antiporter will transport one thing in and one thing out during the transport action. And active transport, there's just all sorts of different kinds of active transport, like so many. There are entire books written about this subject. They can all fall into these categories, but I should note that there are proteins involved in facilitated diffusion that can fall into these categories too. So sometimes it's a little hard to kind of have distinct categories when it comes to this topic. An example of a primary active transport situation is a protein that we have called the sodium-potassium ATPase. This is taken from your textbook. So this is a protein that moves both sodium ions, which are shown in green here, and also potassium ions, which are shown as like these little purple triangles. And so this is the sodium-potassium ATPase, the big purple blob in the cell membrane. And you'll notice ATP here as well. So there's a rule for ATP. So I want you to pay attention to the shape of the protein because it matters. So the sodium ions in this scenario are attached to the inside of the protein, and that then attaches to ATP. So 3 sodiums attach, then ATP attaches, then ATP is hydrolyzed where a phosphate group breaks off the ATP, leaving ADP and a phosphate group, and the phosphate group is now attached to the protein. That attachment triggers a conformation change in this protein so that it closes on the inside of the cell and opens on the outside of the cell, releasing these sodium ions. Once the sodium ions are gone, what we get is, moving down here, notice like this figure goes like kind of in a circle, all right? What happens then is the potassium ions enter and bind, and there's still a phosphate attached at this point in time, and the conformation stays open. And then a moment later, the phosphate detaches, and that triggers a little bit of a conformational change in that the potassium ions like are firmly attached. So the firm attachment of those potassium ions is what triggers the loss of the phosphate. And then the protein is triggered to change shape again, closing on the outside and opening on the inside, and releasing these these potassium ions to the inside, and we're left back able to start the whole scenario again. So there's kind of a gating happening here really, and the gating is due to like the differential binding of ions and phosphates that happens over a series of events. And those events have to happen in a particular order. So it's an ATPase because ATP is being hydrolyzed or broken. That's happening here. And it's called the sodium-potassium ATPase because we're transporting sodium and we're transporting potassium. And you'll note that sodium and potassium both have a +1 charge, and that every time this pump runs, 3 sodiums are being moved outside the cell, and only 2 potassiums are being moved into the cell. So this whole scenario actually creates an electrochemical gradient, and that by having a more charge on the outside, more positive charge on the outside than on the inside. And this is really important for a lot of cell functioning in a variety of different animal tissues, including ourselves. Secondary active transport example is the calcium-sodium antiporter in our heart cells. And so this is an antiporter system. We're going to transport both calcium and sodium using the same protein. One's going out and one's coming in. And it's an active transport system because we're going to hydrolyze ATP, turning it into ADP. And so what will happen is every time this calcium-sodium antiporter system works, part of it's an antiporter and part of it's a uniporter, by the way. I told you these systems are tricky, right? I warned you. So we hydrolyze an ATP, and we pump 2 calciums out, and we bring 1 sodium in. And the accumulation of 2 calcium ions on the outside, especially because they have a 2+ charge and the sodium only has a 1+ charge, you get 4 pluses on the outside and 1 plus on the inside. So we are building an electrochemical gradient by running the calcium sodium antiporter in our heart cells. And the heart will use that electrochemical gradient to transport a number of other molecules, and which we won't get into here, but that's what makes it a secondary active transport system, is this building of an electrochemical gradient that's then harnessed to do other sorts of transport. So I know a lot of this is like more detail than we usually get into in our like introductory bio classes. So I just wanted to leave you with a simple summary, like some absolute take-homes. So really you could think about transport in 3 categories. Simple diffusion, which is down a concentration gradient, and it's just diffusion across the membrane. There's no protein involved. And you could remember that there are chemical criteria for what can simply diffuse across. We often refer to this as simple passive diffusion. Okay. Facilitated diffusion, which is also passive in that you only have chemicals moving down a concentration gradient, but it relies on proteins to facilitate or help that. They could be carrier or channel proteins. A channel protein is shown here. Aquaporin is a great example of this. And then there's active transport, where we've got the movement of chemicals against a concentration gradient. So like there's a chemical moving here, and there's less of it out here than there is in here. Okay? And you're creating often an electrochemical gradient to do that, and you're often using ATP to drive the creation of that electrochemical gradient, and then you might harness it for something else. Okay, so these categories do kind of blend together, but this is some of the ways that you can think about it. And if you're curious about the proton motive force, this isn't a live link, but you could, write it down, and then, or you could search proton motive force and the term citable, S-C-I-T-A-B-L-E, and get to this great reading, which you'll be reading later in this class. Have a great day. Bye.